Bismillahirrahmanirrahim, and welcome back to this next video. And this is another video in the series of videos on the differences between the voltage gated and the ligand gated ion channels. In my previous two videos, I've told you about the ion channels in general, and then I focused on the ligand gated ion channels, and then I give you important examples of the ligand gated ion channels. In this particular part of the video, I'll be focusing on the voltage gated ion channels. So the first thing is what are these voltage gated ion channels? Now these voltage gated ion channels, they are actually the integral membrane proteins that enable the passage of selected ions across the cell membrane. For example, if you are talking about sodium voltage gated ion channels, so that particular ion channel is going to allow the movement of ions across the cell membrane. If you talk about the calcium ion voltage gated ion channels, that means that that particular ion channel is going to allow the movement of the calcium ions across the cell membrane. Now, the major or the most important thing to keep in mind about the voltage gated ion channels that these channels they open and close in response to changes in the transmembrane voltage, which is you can say very much different than the ligand gated ion channels. So in the ligand gated ion channels, when there is a ligand like the neurotransmitter that is going to bind to the ion channel, and because of the binding of that particular ligand, that particular ion channel is going to get open. In this particular case, when you talk about the voltage, the transmembrane voltage that is actually responsible for the opening and the closing of these ion channels, and this play a very key role in the electrical signaling by excitable cells such as the neurons. And I'll give you two very important examples about the neurons, how these voltage gated ion channels they actually work, and how they are going to get open when there is a change in the transmembrane voltage. So the first example that we are going to see in the voltage gated ion channel is the sodium ion channels. Now the sodium ion channels, if you ever have seen the structure of the neuron, so there is an axon. This whole structure that I'm showing you over here, that is the axon. Most of the time these axons they are actually you can say covered by a myelin sheet, and between these myelin sheet there are small openings which are known as the nodes of the Ranvier, and these node of the Ranvier they are actually having the different sort of the ion channels. So in this particular example, I'll be focusing on the voltage gated sodium ion channels which are present at the node of Ranvier in the axon part of a neuron. So this is the whole structure of the you can say an neuronal cells. I'm not going into that because I have a detailed video on that. I'll share the link in the description about the structure of the neuron. So when the signals they are received at the dendroids, and these are actually the dendroids position, these signals they are analyzed by the axon helix. They are transferred to the axon helix, and if the signal is strong enough to cross the threshold value, that signal is going to pass along the axon. What happens is that when the signal that reaches at the axon, that is going to change the transmembrane potential at the axon part over here, especially at the node of Ranvier. And when this you can see this charge, which is actually known as the depolarization, when this depolarization moves along the axon, so the sodium ion channels, which are actually the voltage gated sodium ion channels, they are going to get open because of the changes in the transmembrane potential over here because of this depolarization. And when there is changes in the you can say transmembrane potential, these voltage gated sodium ion channel they are going to get open. So this is one of the important example of the voltage gated sodium ion channels that they open only because of changes in the voltage of the transmembrane, or you can say there are changes in the transmembrane potential. Another important example is the opening of these voltage gated calcium channels. So this is a different sort of channels. It is going to allow the movement of the calcium ion across the neuron. So when we talk about the you can say a structure of the axon terminals, so there are these synaptic vesicles, also known as the neurotransmitter vesicles, and you have got these voltage gated calcium channels. Now initially these voltage gated the voltage gated calcium channels, they are closed, but when the action potential, by that I mean when the transmembrane potential that changes, that is going to move towards the end of the axon, which are known as the axon terminal. This is actually the axon terminal part, and when the action potential it reaches the axon terminal, one of the important thing that is happening at the axon terminal is the opening of these voltage gated calcium channels, because now the voltage there is changing, and when the voltage gated calcium channels they open, they are going to the calcium is going to move inside the axon terminal, and when the calcium that moves inside the axon terminal, that is going to interact with another protein known as the calmodulin. The calcium and the calmodulin, they are going to make a complex, and when this complex is made, it is going to activate another important protein known as the KM kinase 2, and a variety of the reactions they are going to take place after the activation of the KM kinase 2, and ultimately it results in the release of the neurotransmitters, and these neurotransmitters they bind to the receptor present on the postsynaptic neuron. So this is another important example where the ion channel, specifically in this particular case the calcium channels, they are opening because of the changes in the voltage, therefore they are known as the voltage gated calcium channels. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.